Hello, everyone, and thank you for viewing this presentation, where I'll be talking about phenotypic relationships within a quinoa world core collection. My name is Evan Crane. I'm a member of Dr. Kevin Murphy's Sustainable Seed Systems Lab at Washington State University and a PhD candidate in plant breeding and genetics in the Department of Crop and Soil Sciences. I'll first talk about quinoa today in a broader context before defining our research objectives and the study we designed to pursue them. I'll share some preliminary data from the study and talk about the significance of these results in the context of the WSU quinoa breeding program and our international collaborations. Interest in quinoa, driven by the potential to produce exceptionally nutritious seeds under extreme environmental conditions that major crops cannot tolerate, is leading to a worldwide expansion in quinoa production. Broad genetic variability and adaptability resulting from over 7,000 years of domestication, adaptation, diversification, and conservation of genetic resources by Indian farmers has produced a gene pool able to support strategic germplasm development. Tremendous variation exists for plant and inflorescence architectures, grain sizes and colors, and growth habits, in addition to drought, frost, and salinity tolerances. A wide range in seed quality with respect to traits like protein and starch content and composition present myriad possible end uses and the potential to integrate quinoa into global cropping and food systems in a culturally relevant way. Unfortunately, the breadth of quinoa diversity conserved in South America is not available to support global expansion because there is no legal framework to support the sustainable and equitable management of genetic resources. However, a world core collection has been developed by our collaborators at King Abdullah University of Science and Technology in Tuwal, Saudi Arabia. This collection is publicly available and ready to support quinoa global expansion. The collection was developed through opportunistic sampling across quinoa's geographic distribution based on germplasm availability and access. It was originally described as a desperation panel. Given the latitudinal variation in origin among the accessions, there is considerable variation in day length sensitivities. It consists of a non-quinoa group as well as the two primary germplasm pools in quinoa, the lowland and highland groups, and the highland group is further divided into two subgroups. Current efforts are focused on refining the collection using sequencing data to construct a representative panel based on genetic diversity. In a study that I will outline briefly, we set out to accomplish these research objectives. We first wanted to perform an in-depth characterization of the collection, identify sources of variation, and understand relationships between the accessions and the traits. Finally, we aimed to identify the genetic architecture of these traits using a genome-wide association study. And today, I'll just be focusing on the results from our third objective, which I will share with you today. Now, to outline the study design, where I'll focus on the aspects of the study that are most relevant to this presentation. The World Core Collection were planted in a randomized complete block design with 4 blocks in each of 2 greenhouses in May and June of 2018. At harvest, plant height and inflorescence length and width were measured, and a photograph was taken of each inflorescence from each plant. These photographs allowed us to quantify inflorescence morphology traits and also provide a reference image for each plant. Inflorescence morphology traits include inflorescence shape, which is detailed in this card provided by our collaborators at KAUST. Using the harvest photos, we also scored inflorescence density, which is shown here, leafiness, and color. Note the diversity represented here. As I move through these photos, keep in mind that we use them to assess inflorescence morphology in terms of shape, density, leafiness, and color. What I want you to see in these last few photos is evidence of day length sensitivity. These inflorescences can have an elongated primary axis and even show evidence of reverting back to a vegetative state. Note the small leaves present in the apical portion of the inflorescence. Following harvest, the seed samples were moved through our post-harvest phenotyping pipeline, which is shown here, as well as the traits we're able to measure. This photo illustrates some of the diversity in seed traits that we see, and we're particularly interested in assessing protein content and composition, as well as seed size, shape, color, and 1,000 seed weight. Now that you have an outline of the study design and our phenotyping process in mind, I'd like to share some preliminary results from the study. And again, I'll be focusing on the relationships among our traits of interest. To start, I'm going to walk you through this biplot from a principal component analysis. Looking first at the inflorescence traits, we see inflorescence length and width and area positioned together. And interestingly, we see inflorescence leafiness and density opposite, near 1,000 seed weight, seed area, and yield. And these seed traits, looking at them a little more closely, shape is shown here as average eccentricity, which is located near the origin. 1,000 seed weight and seed size are positioned close together and relate to yield. Finally, the seed composition traits estimated using the near-infrared analyzer all grouped together. I have the unit shown here, as well as how we calculate crude protein content using percent nitrogen. And this correlation plot visualizes a Pearson correlation matrix where cells with a black X represent insignificant relationships at a p-value of 0.05. For significant relationships, the correlation coefficient value is color-coded according to the legend shown here. Larger circles represent larger correlation coefficient values, as you can see on the diagonal. And with the time we have, I'll focus on a couple relationships with crude protein fixed at 14% moisture and yield. And I'll start by showing the relationship between crude protein and days to harvest. We found a positive relationship between these traits, which could be a function of additional time to translocate protein into the seed. And our breeding program is focused on early maturing varieties with high protein content. So these accessions are especially relevant to achieving those goals. Before transitioning to the relationships that yield has with these traits, I want to show the relationship between yield and crude protein content. We found a negative relationship, whereas yield increases, crude protein decreases. And while this is the general trend we observed, there are promising instances of varieties that have both high yield and high protein content. Transitioning now to yield, I want to start by showing the relationships between yield and 1,000 seed weight and yield and seed size represented as average area in millimeters squared. We found a positive relationship between yield and 1,000 seed weight, and I want to take a moment to explain how we quantify 1,000 seed weight using our seed scanning system. We start by taking a subsample and then weighing that sample so that we have a known weight, and we capture an image of the subsample using a flatbed scanner. And then we use an algorithm to count the number of seeds in the image in addition to quantifying many other parameters. This algorithm was developed by our collaborator Nathan Miller from the Spalding Lab at University of Wisconsin-Madison, and it's part of a Cybers app and is free to use for academics. We did have a student compare the hand count 1,000 seed weight to the scanner count 1,000 seed weight as part of a small project, and the relationship is strong. The subsampling protocol is not a trivial task, but is definitely faster than manual 1,000 seed counts. To put this in perspective, for this study, if we had not weighed the subsample, we would not have been able to quantify 1,000 seed weight, but we would have saved approximately 120 hours of labor, which at $13 an hour minimum wage in Washington state equates to approximately $1,500 US. I don't even want to know the time and cost analysis for hand counting all of those samples. Large seeded quinoa varieties are typically preferred in the market, So these samples are particularly interesting from that perspective. And when we compare the relationship between yield and seed area to yield and thousand seed weight, there is a slight reduction in the correlation coefficient value. Given this comparison, if we need to increase throughput and reduce cost, we could rely solely on seed size for our assessments. I want to turn now to the relationship between yield and ash content. We found that as ash content increases, yield decreases. We did not necessarily expect to see this, and while we are working to better understand what might be driving this trend, I would appreciate any insight from this community. Finally, I want to show the relationship between yield and days to harvest. Overall, we found a negative correlation between yield and days to harvest. For this figure, I wanted to highlight the samples from the World Core Collection and those from the WSU quinoa variety trials. A major challenge for the WSU quinoa breeding program is to achieve early maturity with the goal of developing germplasm that matures in less than 90 days. Some caveats to keep in mind with this study is that the greenhouse conditions differed from what the plants would be exposed to in the field as part of a typical growing season in Washington state. And while harvested at physiological maturity, the plant condition was not necessarily appropriate for a combine harvest. And as an aside, under short day conditions, we would likely see much different results here. With that being said, out of the 26 entries from the WSU quinoa variety trials, 24 had a mean days to harvest less than 90, which is fantastic to see. What is also exciting to see is that 39 out of the 334 entries from the World Core Collection had a mean days to harvest less than 90. They also met this goal. And this is exciting because these entries have the potential to make a meaningful impact on our breeding program. In summary, I presented a selection of phenotypic relationships that we found within a World Core Collection of quinoa when grown under controlled, long day greenhouse conditions. This collection is available to support the global expansion of quinoa, and this study substantially augments available information on these accessions, which ongoing field trials will complement. These data will be made available in an online database under development at KAUST that breeders will be able to query to identify accessions with desirable characteristics. The core collection is being grown out as part of a wonderful international collaboration that we are fortunate to be a part of, and the generous distribution of this germplasm has made several international projects possible within our own research group. With that, I would like to thank the collaborators and lab members that have contributed to this work, especially Mark Tester for his leadership in developing the panel and sharing it with such generosity. For more information on our lab, please visit our website or connect with us on Facebook and Instagram. On our YouTube channel, you can find many quinoa presentations, including one on the development of the post-harvest phenotyping pipeline. And here's a short video where you can see that in action. You can scan these QR codes using your phone camera to link to them directly. Thank you to our funding sources for their support and to the organizers for the opportunity to present. Before I conclude, I want to acknowledge and extend many thanks to the Indigenous communities that have safeguarded these genetic resources and highlight the need for the international quinoa community to respect their stewardship and commitment to this crop with our work. Thank you for your attention.